ma'am, within 30 seconds we will start. We are live now.
So, shall we proceed?
Yes, we are live now. We can start.
Okay. Sure.
Bye.
So, hello everyone. This is Darini Chokhadia, the event manager and host for today's live podcast, The Next Experiment by the Scicomm Synapse, a podcast featuring researchers and scientists. I am delighted to welcome our honorable guest and speaker, Dr. Nupoor Chowdhary. And today we will be in conversation with her on why bioinformatics matters and where to begin. But before that, I would like to introduce a bit of Scicomm Synapse. At Scicomm Synapse, we strive to be a bridge between the world of research and the curious minds in our community. It's a space where ideas ignite, questions flourish, and discoveries inspire change. Guided by our belief in thinking, growing, and sharing, our mission is simple: to bring science closer to people in a way that's meaningful and inspiring. Now, I would like to introduce Dr. Nupur Chowdhary. Dr. Nupur Chaudhary is an assistant professor in the Department of Microbiology and Biotechnology Center, Faculty of Science at the Maharaja Sayajirao University of Baroda, MS University. She holds a master's degree in bioinformatics from Banasthali Vidyapith, Rajasthan, and PhD in bioinformatics from Anna University, Chennai. A recipient of the prestigious DST Women Scientist Grant from the Government of India, she has strong research expertise in genomics, transcriptomics, computer-aided drug design, systems biology, and genome-scale metabolic modeling. She has published her research in various reputed national and international journals and is engaged in academic monitoring. In addition to her academic roles, she is actively involved in research and provides computational support for genomic and transcriptomics studies, underpinning ongoing research on antifungal drug resistance and pathogenic Fusarium solani in Professor Devarshi Dudge's laboratory at the MS University, Baroda. So thank you, Nopur ma'am. So we shall now proceed towards the question answer sessions to explore your insights in more depth. So shall we?
Yeah, sure. Thank you so much, Dharani, for such a wonderful introduction. And thank you to the entire team of Scicomm Synapse for inviting me and to be live on this platform. Thank you so much.
You're welcome, ma'am. So the first question is, you began your academic journey in chemistry and later moved into the bioinformatics and genomics. What motivated this transition, and how did you navigate the learning curve?
So to start with, I did my master's in 2005 and 2007 batch. So at that time, biotechnology was at its peak in India. But talking about bioinformatics, it was still in a very new and emerging field. Right? So very few Indian universities were offering bioinformatics as a formal course. And I'm talking about nearly 20 years ago, right? So one day I came across a newspaper advertisement for admissions at Banasthali Vidyapith, Rajasthan. And eventually I received admission offers in 3 courses: pharmaceutical chemistry, biotechnology and microbiology, and bioinformatics. So when I discussed about bioinformatics with my faculties at that time, even they were clueless. They were not clear about the field. The only explanation I could get was that it involved using computational or computers in biology in some way. So my shift from chemistry to bioinformatics was, you can say, largely driven by curiosity rather than a clearly defined career plan. So as a 20-20-year-old, the idea, it felt fascinating, and it seemed like a unique opportunity to learn computer science and coding while coming from a chemistry or a biology background, something that was almost unimaginable at back then. So I felt like I could become a bio IT person without doing a formal BTech in computer science. And talking about the learning curve, so definitely it was challenging since I come from a non-programming background. So concepts like coding and statistics were quite intimidating initially. But in the first semester, it was extremely difficult to understand the algorithms behind, even the basic bioinformatics tools like BLAST, multiple sequence alignment, or concepts such as PAM and BLOSSOM matrices, not only for me but for the entire class. So we had to put a lot of extra effort, but gradually the things started to fall into place. And but I would say we had to read a lot to understand those basic concepts. And but that early step into an unfamiliar field actually taught me to stay curious and keep learning and not to be afraid of trying something new.
So that's really empowering, and that's really insightful. Your journey actually shows how curiosity can beautifully bridge discipline.
Exactly.
So, heading on to the next question, your PhD work focused on genome re-annotation and metabolic modeling for biohydrogen production. What was the moment when you realized computational biology could drive real world solutions?
Okay. Um, so, in our time also, we used to have the summer internships and dissertation project, right, in the last 6 months of our entire master's course. So the work in that, during your dissertation and internship work, the work is often divided into smaller, well-defined targets. You are usually assigned a specific work of a larger lab project, and at that stage, the focus is mainly on learning a few tools or acquiring a specific programming skill, right? So the real work start during a PhD, जब आप real biological problems पे काम करते हो, and then you make a pipeline on how to achieve or how to prove your hypothesis. So while working on genome scale reannotation for biohydrogen producing organisms, I realized that existing annotations were often incomplete. If you go and check NCBI and download a particular genome and see that, you will find out almost 40 to around 40% of the genes are still not annotated. They are still hypothetical, so putative in nature. And at that time also, when I started with my PhD work, it I found out that annotations were still incomplete, and it directly affected how metabolic pathways were interpreted. So once we began reannotating the entire gene, it became clear that manual analysis was impossible. So we were doing with large data sets, fetching functional prediction from multiple tools, performing statistical analysis. So this forced us to automate the entire workflow. And actually, after reannotating the whole genome, we were able to create a genome scale model and see how the changes at gene level altered the pathway fluxes and finally influenced the hydrogen production. We checked the gene knockouts computationally. So that was the moment I realized that computational biology wasn't just about analyzing the data, it could actually generate testable hypotheses and guide real biological and industrial outcomes. So I would say, like, instead of performing countless trial and error experiments, we could use computational models to predict bottlenecks, identify the key enzymes, and prioritize our targets for optimization. So that ability will reduce your experimental time and cost while improving biological understandings. And it made it clear to me that computational biology can drive this, the real world solutions. And because of that only, the computational approaches such as molecular docking or virtual screening or dynamic simulation, and other in silico methods are now routinely integrated into drug discovery workflows within the within the pharmaceutical industries. So yeah.
Yeah, so that's really fascinating. It's really fascinating to hear. It's inspiring how computation translated into real world impacts in your work.
Exactly.
Yeah. So heading towards the next question, during your DHT Woman Scientist Fellowship, you led a project as a principal investigator. How did the experience shape your confidence as a scientist?
Mm, So I would say that that was a very important phase in my career, because for the first time I was working as a principal investigator. I was involved in every aspect, from defining the research, then questions, and designing the computational strategy, to managing the timelines, the objectives to finish at a particular timeline, because you have to give your project reports, you have to submit every in the second year of your DST Women Scientist Fellowship. So leading the project, it actually strengthened my leadership skill. I actually learned how to communicate the ideas clearly. So one experience during the DST Women Scientist Fellowship interview, which I would like to share at on this platform, is like during the interview, before they grant the funding, of course, uh, they will call you for an interview, and that you have to present your work in front of the panel members. So when I was presenting my project in front of the panel members, who are actually the highly renowned scientists from different institutions, so as I spoke, I noticed that their faces were completely neutral. There was no nods, no smiles, no visible reaction. So unconsciously, I expected some kinds of visible validation from the panel, right? But at that moment, I started doubting myself. I finished my presentation with confidence, but internally, I felt demotivated, and assumed that I probably would not receive the funding. So, but however, the outcome was very different. I was awarded the prestigious Women Scientist funding, and that experience taught me that senior researchers often evaluate your work very objectively, and their lack of visible reaction doesn't mean that they are unimpressed. What matters is the clarity of your thinking, the rigor of your work, and how confidently you present it. So, it taught me actually that to trust my preparation and stand by my science and not get demotivated, right?
Yeah, so this is a really important aspect in life.
Exactly.
Like, whenever you are, surrounded by challenges, so the best thing is to be motivated and be strong in order to win the world. So yeah,
Exactly.
that's
You have
absolutely.
to be motivated. You have
Yeah.
to confidently present your work.
So that's really empowering. Your experience as a principal investigator clearly shaped your leadership journeys. With that in mind, let's go towards the next question. You have worked across diverse areas from transcriptomics and systems biology to drug discovery and sustainability. How important has the interdisciplinarity been in shaping your research identity?
Okay. First of all, I would say that, these areas, the transcriptomics or systems biology, or you may say genomic study, they are all interlinked. You know, like for my research work, like I use genomics and systems biology. It works side by side. Genomics provide you the large data sets, and it helps you to annotate the genes. And systems biology uses that information to build the genome scale network models. So these models can then be analyzed for different approaches like flux balance analysis, or to understand how the systems behave as a whole, rather than how genes works together and not in isolation, right? So you are studying the entire system as a whole. Similarly, systems biology will provide, since it is providing the complete view of how the pathways are interlinked, they will help you identify the key components and interaction in the disease. So once we understand which pathway and network components are crucial in disease, we can identify the potential drug targets, and the study that will help prioritize which target are the most promising one are based again on both genomics and systems biology data. Right. So in the current scenario where we are talking about large data sets, complex diseases, and multi-omics approaches, these are the new normal. Interdisciplinary is no longer optional. It's essential. It helps us to see a bigger picture. It helps you to ask better questions and generate results that are both meaningful and useful across different scientific communities. So you have to be open to multiple discipline, and that made me a more adaptable researcher, and maybe a more impactful researcher.
Yeah, so I actually love how the highlights and the power of interdisciplinarity in modern science shaped your identity. Heading to the next question, you returned to academia as a faculty member after extensive research experience. How did your perspective on teaching and mentoring change after years of hands-on research?
So I always liked teaching, but this years of experience in research, it changed the way I teach. It changed significantly because we were deeply involved in designing pipelines, troubleshooting the field analysis, and working with the real biological data. I began to see teaching as a process of helping the students how to learn how to think, and not like just to learn what to learn. So research taught me that concepts only make sense when students understand their application. So now if I'm teaching them, so now when I teach, I consciously connect the theory to the real research problems. So what method, why the method has been used, what assumptions it makes, and what are its limitations. So I try to bring my own research experience into the classroom. So suppose if I'm teaching statistics, biostatistics, so I don't treat it as a purely as a subject. I explain how actually I used it to analyze uh, uh, statistics, to analyze my whole genome reannotation data, how statistics helped me decide which results were meaningful, which were noise, and how to draw the reliable biological conclusions from it. So with research experience, I feel we can better prepare students for the future, whether for PhD interviews, industry roles, or independent research. So by sharing, you have to share them your the real world expectations, and you have to teach them the problem solving skills. And that's what research teaches us. So I try to deliver the same thing in my class during my lectures.
So this perspective is really valuable on basically how teaching is valuable. Plus, it adds research experience that truly adds depth to the mentorship. And
I see, yes. And I feel most of the teachers are using the same protocol these days, same concept these days.
Yeah, absolutely. So heading towards the next question, as a woman scientist who has navigated funding, research gaps, and career continuity, what structural challenges did you encounter, and what helped you persist?
I would say, women scientists in STEM, and this question is really very close to my heart. So being a women scientist in STEM, it comes with unique challenges, right? Especially when it comes to career continuity and personal responsibilities. So, but for a women scientist, I strongly believe challenges are more at personal front. At some point of time, every woman has to make difficult choices for them, either being between career and family or constantly trying to manage both. right? So we often use the word balance, but in reality, it is never a perfect balance. At different stages of life, you end up sacrificing something. So for me, the biggest challenge began when I became a mother. I became a mother even before starting my PhD, and that guilt that comes with motherhood, especially not being able to give enough time to a child, stays with you for years. So I still remember leaving my child at daycare, crying when I went to lab, and that is one of the hardest moments in the woman's life, right? Then you have, you have guilt that you have to make professional choices also. I had to let go some of my postdoctoral opportunities abroad because I didn't wanted my child to stay away from his father. And those decisions were not easy. And that will slow down your certain career leap. So that guilt remains, but they are not but they were necessary at that stage of my life. But you know people say that uh, a success behind a successful man there is a woman, but I truly believe that behind every successful woman, I may not say that I'm very successful, but whatever small success I have achieved, there is an entire support system. In my case, this included my family, my friends, in my society, and my mentors. So my child got adapted to my schedule. My husband was a constant support, source of strength. And I still remember my mother coming and staying with me for months, while my father, who was over 55 at that time, he managed his alone in his hometown, taking care of his meals and daily needs so that I can pursue my dream. And even today, this support system continues. My younger child is just 6 years old, and on the day when the caretaker is on leave, or when she's not around, it's often my friends' mothers who step in. So challenges are there, but the trust and support I receive from people around me. It allow me to focus on my work and with peace of mind, and that is what helped me to persist. you know so the same advice I give to all the young researchers, especially to girls, to the women researchers, young women researchers. So I've seen many girls after a certain period of time, especially after becoming a mother, they would compromise on their career due to the certain challenges in their life. So I would say take a break if necessary, but be ready to make a comeback. Never hesitate to ask for help. You don't have to do everything on your own. You don't have to be a superwoman all the time. There's nothing wrong in leaning on others when you need support. Asking for help doesn't make you weak. I would say trust yourself, accept support, and move forward at your own pace.
Absolutely, because coming to the the phase, the postpartum, and postpartum challenges are really really more. And coming to that, I believe that this where that time when we receive support and when we receive guidance, I think that helps us coming back to our career and coming as a strong comeback, right? So
Exactly.
I think this is really honest perspective, and this could not only help the women scientists, but it can also help all the women who are passing through the same phase.
Exactly.
Yeah,
Exactly.
yeah. So thank you for this wonderful insight. And looking back at your journey so far, what advice would you give to students who are interested in bioinformatics but come from non-computational background?
Okay, so I have made a small presentation on this topic itself, but still, I would like say a few words before we start with the presentation. Uh, my first advice to students from the non-computational background will be don't be intimidated by bioinformatics. You don't need to become a bioinformatics expert overnight. Start small and focus on understanding the logic behind the tools rather than memorizing the command. They'll write all the commands and they won't understand why we are running those commands. So this is the thing I face. So I'm seeing in the young students who are not from computational background. So it's important to learn how to troubleshoot when you are stuck. But you know, I have often seen students, they give up on a problem simply because they encounter errors while running a tool. But however, errors are a normal part of computational work, right? And learning how to interpret and fix them is crucial skill in bioinformatics. So be patient with yourself. What matters is consistency, not speed. So be consistent. And there are many online workshops that are available that teaches different bioinformatics tools and pipelines. So depending on your area of research, you have to identify the right ones before registering. And a good workshop would not be showing you how to run the commands, but it will also explain you the logic behind running that command and the algorithms used by the tools. So otherwise, what will happen at the end of the day, you will learn how to execute the tool without truly understanding it. And if you are stuck in between, if you have any error in between, you will not be able to how to troubleshoot that error. So once you register for those programs, it is important to attend the sessions regularly. Mind my words, it is important to attend the sessions regularly. Many participants I have seen, they assume that the recordings will be shared and they can watch them later. But bioinformatics does not work that way. When you run a tool or pipeline for the first time, you are almost guaranteed to encounter errors. So if you are attending the live sessions, uh, you can immediately ask the questions and resolve the issue. So if it will be kind of a 1 hour session, you can attend the session, you can learn the tool, you can learn the commands you can run, and then before the next session that is going to happen the next day, you can run and be prepared with all the commands. So if you encounter any error in between, you'll be able to ask the question the next day. You can ask the question in the next session, and that will make a huge difference in building that confidence. It can you know so I would strongly recommend or encourage the students to build strong fundamentals first. Tools will keep changing, but these fundamentals will stay with you forever in your career. So attend the sessions, run the commands before the next session. And if you are stuck somewhere, ask the instructor, and that issue will be resolved there in the online session. But if you're not attending such session, if you're waiting, okay, theek hai, we'll be getting the videos. You're going to get the videos recordings after that. And if you're stuck somewhere, whom you're going to ask, right? So if you are attending some session, attend it regularly and run the tool. Don't only attend. Whatever you have learned that day, run after the session gets over and be prepared for the next session. Otherwise, if you're not attending it, you will be stuck somewhere and then you will give up.
Yeah, so I think this is a perfect note for students, especially the scientists, and I think especially for becoming a scientist, you need to be consistent, and for that, I think you need to clear your doubts and questions and all, because that is going to build up a base. I mean, the strong base for which you can build up your career. So I think there should not be any shyness for asking anything. And those students who are stuck in between should definitely revisit the topics and even ask the mentors for a better scientific career. So yeah, ma'am. And yeah, so that's really good. And I would now like to hand over your slide presentation. So yeah, just go ahead.
Yeah, okay. Give me a moment. I'll share my screen.
Sure, ma'am.
Are you able to see my screen now?
Yes, ma'am. Ma'am, can you make it please full screen?
Yeah.
Yeah, it's visible, ma'am.
Okay. Okay. So the topic of today's presentation would be from wet lab to workstations, right? So the first thing is like why there is a growing importance of computational skills in life sciences. It's all because like one experiment today can generate more data than we can analyze manually. Is my voice audible?
So it's audible. It's audible.
Okay, thank you, Samir. So like we are getting exploded with a large amount of data, massive data on a daily basis. And almost every biological experiment now ends as a computer readable file. We get the DNA sequenced in FASTQ, FASTA files, SAM or BAM files. So understanding of these type of files output are important. And even for RNA-seq data, you get millions of reads, but unless we clean the data, we map it and analyze it properly, we cannot say which genes are active or altered or biologically important. So you get the results in the form of count matrices. And from those count matrices, you really need to understand the terms like p-value, fold change, etc., to actually understand the differentially expressed genes. So for these things, there is a growing need of computational skills. Even today, most high impact research is interdisciplinary in nature. So integrating multiple disciplines increases the scope of getting published, the depth and the novelty of the study, which in turn improves its chances of publication in high impact journals. So why there is a data explosion in biology? So over the last few years, the cost of sequencing has dropped. So what earlier used to be expensive and limited to a few labs is now widely accessible. So as a result, biological research has entered an era of massive data generation. A single experiment can produce enormous amount of data, whether it is a whole genome sequencing, RNA sequencing, proteomics, or clinical or imaging data. So, for example, like whole genome sequencing now generates billions of base pair in a single run, and your RNA sequence experiment will produce millions of reads for each sample. So while generating data has become easier and faster, making the making sense of this data is real challenge. Because raw data on its own has no biological meaning unless it is properly processed, analyzed, and interpreted. So, why computational biology skills are no longer optional? So, first, because biology today is no longer about studying one gene or one protein at a time. Most modern studies involve multiple layers of data: genomics, transcriptomics, proteomics, metabolomics. So, to integrate and interpret all these layers together, computational tools are absolutely necessary. Without computation, these data sets remain disconnected and incomplete. Secondly, AI has transformed how we approach the biological structures. Tools like AlphaFold has made it possible to predict protein structures, but using these tools meaningfully, it requires computational understanding. Knowing how to run them, access without access with access the confidence scores that they generate, and interpret the biological relevance of the prediction. Then in systems biology, in which I worked during my PhD, where the focus is on understanding networks. So how genes, proteins, the reactions, and the pathways, they interact with each other. So these are complex dynamic systems, and only way to study them is through computational modeling and simulation. And the fourth one, of course, reproducibility and pipelines. Computational pipelines, it ensures that the analysis can be repeatable. If you generate, suppose if you have a whole genome data or a whole transcriptome or a transcriptome data with you, and you want to perform a certain kind of analysis with that data, you can actually generate a pipeline. And these pipelines are reproducible. If you want to do a similar kind of analysis on a different data set, you can just import your data set and run the same pipeline, and finally you're going to get the results. So, this is especially important when dealing with large data sets where manual analysis is, not practical nor reliable, and you are repeatedly doing a same kind of analysis on different data sets. Then making a pipeline and using it repeatedly, it's computational skills makes it easier. Yeah, so there's no foundation who can enter this field. If you're curious, if you're patient, and if you're willing to learn, you belong to this place. You belong to this field. Any biotechnology and microbiology, if either you're from life sciences, molecular biology, chemistry, biochemistry, even if you're from physics, maths, computer science, or pharmaceutical sciences, your background there, that is not going to restrict you. It's just that your curiosity will define you, So the field welcomes anyone willing to bridge experiment and the computational thinking. Coming to wet lab versus computational biology, not a rivalry, it's a partnership. I hope many of you will agree with me. They would agree. So wet lab experiments are where biological questions originate, right? Observations in the labs help us form the hypothesis, test them under control condition. And on the other hand, the computational biology brings scale and the speed to the analysis. It allows us to analyze thousands of samples at once, explore multiple possibilities quickly, and even even predict outcomes before we invest time and resources into expensive experiments. So the real power comes when these two approaches are integrated. Computational analysis can suggest the most promising genes, pathways, or drug targets. Wet lab experiments then validate these predictions, and the experimental results are again fed back into the computational models, making them more accurate. And this actually creates a continuous cycle of discovery. So there should always be a partnership between wet lab and computational biology work to generate meaningful data. So I have just dotted down few points over here, like on which area the computational biologists mostly work, but not restricted to only these points that I have dotted here. So computational biologists they work on different fields, like they analyze sequencing data, they process the raw genomic, transcriptomics, or the epigenomic data through high quality alignment, variant calling pipelines to identify meaningful biological signals. Then they study the disease mechanisms using different approaches like comparative analysis and pathway modeling, to how the genetic variants or gene expression changes or the protein modifications contribute to the disease state. Then they are also working in predicting the protein structures using deep learning models, deep learning tools, and they predict how the protein folds. They identify their binding sites. They understand the structure function relationships that drive the biological activity. And they also assist in drug discovery process. They screen compounds, the libraries virtually, They predict the drug targets interactions, and they identify the potential therapeutic candidates before expensive and experimental validation. And they also build analysis pipeline. They create reproducible, as I earlier said, documented workflows that automate the repetitive tasks, and that pipeline that can be shared also on different websites, on different locations, and it will enable the others to use their methods. So yeah, the common myths before breaking down the fear. So the common myths are like, I must be an expert coder, or only computer science students they succeed, and I'm too late to start. No, nobody is too late to start. Everyone struggles at the beginning, so you can start at any point. But the main thing is you should start, right? And the learning is gradual. Nobody will become a proficient overnight. And biology knowledge is actually powerful. If you are from a biology background, if you are from microbiology, biotechnology, biochemistry background, chemistry background, that will actually increase it, that will actually help you. So every bioinformatics analysis, it begins with a biological problem, such as understanding why a gene is differentially expressed, which pathway is altered in a disease, or which protein could serve as a potential drug target. So without a strong biological foundation, it is easy to run the computation analysis without clearly knowing what question is being addressed. Biology is actually crucial for the correct interpretation of results, because computational tools will typically produce the outputs, right? Like gene list, it will give you the pathways, it will give you the interaction networks or docking scores, but the biology knowledge insights will help determine whether these results make sense or not, whether it aligns with known mechanism, or these are this could simply be an artifact or a noise. So I would say, in addition, biological understanding guides the choice of appropriate method and parameters, such as selecting the right reference, selecting the right genomes, databases, and giving the right thresholds while running a particular bioinformatics pipeline or an analysis. So what I would advise to master's student is, those who are doing your master's in different field. So your master's program is the ideal time to build a computational foundation. Right? So during a master's program, I would advise them to learn the basics of Linux, because most of the tools run on Linux system. Then at least you should learn one programming language, either R or Python. You should learn one omics workflow, either whole genomics or transcriptomics. Excuse me. Understanding one workflow will help you to learn others, and it will actually it will ease you out, and you can learn other pipelines easily. Then you should familiarize yourself with how machine learning applies to biology. And of course, you don't have to build models from scratch at this age, but understanding them, how to use the AI tools, is important. Then literature reading skills. You should learn to critically read the method section, understand the analytical approaches in the papers, and recognize what computational methods and when to use those computational methods, and whether those computational methods are appropriate or flawed. So this is the basic roadmap we try to follow at the MS University during the master's program practicals, so that students systematically learn the essential skills in computational biology before they graduate. But if for other students, if this option is not available in their university, or for the research students who are doing their who are doing their PhD, as I told earlier, they can identify the correct workshops as per their area of research and attend that. Yeah. So you can have, the computational skills unlocks diverse opportunities. You can enter into PhD program. You can enter into industry roles. So I've, even during PhD interviews, I have learned. I've learned that many PIs, they are looking for students, scholars who have an experience, a basic experience in computational biology during their master's program. So actually that increases the chance of you being selected in that lab. Then you can join several research lab at JRF or bioinformatician support. You can have different global opportunities. You can enter into pharma industries, and you can combine machine learning with therapeutics development and precision medicines. And I would say, even if you're confused, start early in your program, practice weekly, follow one clear roadmap, pick one structured learning path or tutorial series. Avoid jumping between resources, because depth will matter initially. Don't fear errors. Error messages are learning opportunities, not failures. So you should learn how to troubleshoot. Then ask questions actively. Use online communities. We have huge online communities of computational biologists. You can talk to your mentors, you can talk to your peers, you can mail me as well, and you can approach computational researchers. Everyone has started as a beginner, right? So this field needs more people like you, biologists who understand both experimental and computational sides of modern biology. So your journey starts with a single command line. Take the first step today. Yeah. So this is the last slide. Actually, this slide I show to all the my master student. So in this slide, you can see three scientists: David Baker, Dermis Hassabis, and John Jumper. They recently won the Chemistry Nobel Prize for creating amazing computational tools that can predict and design the protein structures. And they received the Nobel Prize in 2024. I mean, this shows that with computational biology, you can solve the real world problems faster, and that even open doors to discoveries that were once, thought to be impossible. So if you're curious, this field is full of opportunities for you to make a real impact. So thank you. So with this, I end up my slide with my presentation, and
Okay,
yeah, I'm open to
Go.
questions now.
Yeah. So now basically, this was a wonderful presentation, ma'am. It was truly enlightening. So now I would love to hear from our audience. This is your time. Please go ahead and share your questions. Somalia, please take the stage. You might have those questions with you.
So just a minute. Give me a minute. So, yeah. 108 answer is 108 answer is. Just a second. Give me 1 minute. So, ma'am, we have a very great. It was really an intriguing talk, and we have a list of questions.
Oh
So
my God.
that
Yeah.
completely I cannot take, but the some
But
of
yeah,
the
you can take some
questions
of the important questions. Yeah.
that I will be taking on. So, ma'am, the very first question is a very basic question from some clear concepts. He is a person on our YouTube. So
Yeah.
he asked that if we want to do course on bioinformatics from any app like Coursera, Swayam, in which particular course related to bioinformatics should we enroll, and will it be worthy to do such courses?
Yeah, that is the thing I already mentioned. So depending on your research area, or if you are a master student, you can do basic courses on uh, molecular docking, or how to use bioinformatics basic tools. For you can learn the basic tools from these workshops. So you should enroll for such type of courses. And if you are enrolling for such type of courses, I would again repeat, attend those courses when they are going live. If you are just okay that we will get the video recordings and we can learn later, then you will end up doing mistakes, and errors will be there generated because you will be doing it for the first time, and then nobody will be there to help you, right? So you'll be stuck in between. And for researcher, depending on your research work, you can do. There are many even we from our department, we have started with a few workshops on different topics, and we'll be doing it every semester, maybe online. And apart from us, there are other private organizations also who are doing you are conducting such kind of workshops on a regular basis. So you can just google it and find out, and even on LinkedIn you can see those posts. So
Yeah.
depending on your interest, you can join any of the courses.
Yes, ma'am. So next question
Start
is
with basics. Start with basics. Yeah.
So, ma'am, next question is by Abhidanand. It's a very important question. So, she asked that, ma'am, how one can access resources to related to bioinformatics like simulation tools, because it is not possible that every institute have the facility, or they won't allow to access from other institute as well. Ma'am, it's a really intriguing thing.
Yeah, that is the thing. But then you have to make connections. You'll have to collaborate with somebody, because not all the university, not all the institutions will have that high-end workstations, high-end servers where you can run the MD simulations. So in such cases, I would suggest that you should have your PI, or under whom you're doing your PhD, they should collaborate with people. People are there to help. People are there to help. You have to mail them, you have to collaborate with them, and then they will allow access to work on these tools.
Yeah, that's really
Yeah,
great.
yeah,
Collab
exactly,
the networking is the key for here.
exactly. Especially in bioinformatics, I would feel, because there are so many high-end tools that can be run only on high-end servers. So you need to collaborate. They will give you access to their servers, and you can either you can run from your end, or you can visit their lab and they will teach you there.
Yeah, so we can say that your network is your net worth.
Exactly, exactly, exactly.
And second question is that, does it makes a lab research work impactful enough without wet lab to get a published in Q1 or Q2 journals?
Yeah, of course, I have published. I have published in PLOS, but the thing is that analysis should be in depth, and there you to prove a thing, prove a hypothesis, you have to do a very in-depth analysis. You have to use many tools, and you have to analyze the data from those tools. uh, there should be no noise. The data that you get from those tools should be impactful. The results that you get from those tools should be impactful. And you have to statistically prove also. You have to statistically also prove the results. If those results are actually correct, right? So yeah, you can publish data with wet lab analysis.
So,
Sorry, with
ma'am,
dry lab analysis.
questions are not ending, but we will take two or three more questions. So, ma'am, the people are asking that they learn how to run the commands and all through workshops and all, but they don't get after they are pro in the commands, but they don't know how to apply it in real world biological problems. So suppose we learn the R command, we learn the Python command, but how should we understand that where to apply it and how to apply it in real world biological problem?
See, That's what I'm saying. Ki you have to choose a workshop that is not only telling you what commands you have to run, but that actually teaches you where to run that command and when to use that command in the
Yeah,
real
ma'am,
world
that,
scenario, real world problems. Yeah.
are there such kind of workshops available which
Yeah, of course,
tells?
that's what I'm saying. You have to select. You have to, before before actually enrolling for any workshop, you should connect with them. You should ask them what they are going to, what sort of sessions you are going to have, and whether you are going to get some knowledge from that or not. Whether the algorithm behind the tools or the reason why we are running those commands are being will be covered in the sessions or not. Otherwise, there is no use of attending those workshops, right?
Yeah. So, ma'am, last question we will be taking in the audience question round. So,
Sure.
ma'am, in your career till now, solving what biological problem using computational bio has been your favorite discovery?
Yeah. So I would say when I was working with my when I was working uh, during my PhD, when I generated a whole genome model for my organism. I was working on an organism for biohydrogen production to enhance biohydrogen production in that particular bacteria. And we generate a whole genome model to see what uh, genes could be knocked out to increase the hydrogen production. So we have to make the entire pathway. You have to generate the entire pathways. You have to study all the pathways that are interlinked, and that was actually very difficult to create a whole genome model, to run metabolic fluxes on that, and to see that actually the production in hydrogen, the hydrogen production or the flux towards hydrogen is increased by computational predicting certain gene knockouts. So that was the most interesting thing that I have done with this computational biology knowledge.
Yeah, ma'am. Ma'am, I see a lot of people are interested in learning bioinformatics,
Yeah.
and yeah, and they are asking, can we get an email address of yours?
Yeah, sure. You can share my email address,
Sure.
and I'll be more than happy to help everyone, each and one out there who are interested in learning bioinformatics.
So, ma'am, even I am interested in learning bioinformatics, and I am already a student of yours.
Of
In
course, you are my student. You were my student. Yeah.
yeah, so ma'am, it would be really great if we can do a series of workshop kind of thing for the students who are highly interested and highly dedicated to learn, like which will include some practical session and assignments as well, if possible.
Yeah, okay, we can look forward into that.
Yeah.
We'll see to that how we
Yeah.
can work.
Yeah. So, ma'am, thank you so much, ma'am, for addressing all those questions. And I have my questions as well. Maybe I will address it later on. Yeah, because we are running a bit short of the time, and the
Exactly.
session was really intriguing. So, I request Dharini, it would be really helpful if you can continue the event. Yeah.
Sure. Yeah. So, first of all, I would like to deliver a vote of thanks to Dr. Nupoor. As we conclude today's session, I would like to sincerely thank our speaker for such an insightful and inspiring journey with us. Your experience from transitioning across disciplines to leading impactful research have offered valuable lessons for our students and researchers alike. Your reflections on interdisciplinarity, leadership, teaching, and resiliency, especially a woman scientist, were truly empowering. The advice you shared for students entering bioinformatics from non computational background will surely encourage many listeners. Also, to our audience, on behalf of Scicomm Synapse, thank you once again for your time and thoughtful insights. It's been a pleasure hosting you for today's event. We would really appreciate if you can take a moment to really share your feedback for our next event to make it a little better and energized one. There's a link for feedback form in the comments to receive e-certificate. Please like, share, and subscribe for our YouTube channel for more information. Do visit www.thepsychomsynapse.in. Until then, think, grow, and share.
So before we wind up,
Sure.
end the session, I would really want to thank the entire team of Scicomm Synapse again, to Dharya, Ajinkya, Soumilya, Dharini, everyone, for giving me this platform. And I'm so happy that so many people have joined, and so they were online, and they were so curious to learn about bioinformatics, and uh, how in future they have so many questions. So I would love to answer all the questions via email, and they can connect with me. They can email me their questions. They can come to our university, to our department, whenever they are in Baroda. So you all are most welcome. And thanks to all of you for giving me this platform. And yeah, thank you. And
Thank you
such a
so much.
well spent actually.
Yeah, actually, this was really inspiring, inspirational, as well as informative. So I think, for anyone who is doing research or into any other sector, this session has been really, really helpful to them, as well as, this was kind of boost pack, like, whenever you are surrounded by challenges and everything. I think it is very important to have your self confidence, as well as you can take a moment to pause for a moment. Then I think this is going to be really what I can say, very demotivating for an individual. So yeah, I'm really, really impressed by the way you have shared it, and I'm really a good fan of yours now.
Thank you, Tarini. And that's what I would advise everyone, not to stop in between. You have to troubleshoot the errors. You have many online communities, you have many computational biologists out there. You can seek help from them, but don't stop.
Yeah, absolutely.
I would like to inform you one great thing. So as soon as we published your webinar, your information regarding your webinar on our website, our
Mhm.
page views has been increased by 5100 percentage within one day.
Oh my God. Thank you so much. So, well, yeah, thanks to all the viewers out there. Yeah.
The unique visitors on our website, they have been increased by 2100 percentage.
And all credit to goes to all of you, to all the organizers, right? For connecting with me. Yeah.
In the live, then only I can see the what's the status on YouTube. That, but I have seen that there are around 20 to 25 constant viewers which have been on our YouTube session in this current
Great.
webinar.
That's great.
Yeah,
All
I'm really,
credit goes to the entire team of Scicomm Synapse. Thank you.
yeah. Thank you so much, ma'am. Yeah,
Thank you. So
thank you.
can we wind up for today?
Yeah, for sure, ma'am. Yeah.
Thank you.
Have a
And
great day. Have a great day, everyone.
great day to all of you as well. Thank you.
Thank you, ma'am.